Welcome, everyone. I'm excited to kick off our presentation today on plant growth and development. This is the first slide, so I just want to warmly welcome you all and introduce the topic. Today, we'll explore how plants grow, develop, and what factors influence their growth processes. We'll cover key stages, mechanisms, and some fascinating aspects of plant biology. So, let's get started and dive into the wonderful world of plant growth. Now, let's move on to slide 2, which covers the topic of photoperiodism. Photoperiodism is essentially how plants respond to the length of day and night, and it plays a crucial role in controlling flowering and other developmental processes. Understanding this helps us grasp how plants adapt to their environment and time their growth cycles. It's interconnected with development, growth, and even how plants regulate their internal activities through various regulators. This slide is part of a broader discussion on plant development, emphasizing how external cues like light influence internal processes. So, as we explore this, keep in mind how photoperiodism fits into the overall picture of plant growth and adaptation. Welcome to the introduction. On this slide, I want to emphasize the importance of setting the stage for our discussion. We'll cover the key points thoroughly, but in a conversational way, making sure the ideas are clear and engaging. It's essential to highlight the most important concepts so they stand out and resonate with you. As we go through, I'll connect these points to the overall theme of the presentation, ensuring everything ties together smoothly. This introduction is about creating a solid foundation for what's to come, so let's keep it focused and insightful. So all cells of a plant develop from a single starting point, the zygote. This is the very first cell formed when a sperm fertilizes an egg. From that zygote, a multitude of cells are produced. These cells then organize themselves into different tissues and organs, each with specific functions crucial for the plant's survival. Development, in this context, is really about two main processes: growth and differentiation. Growth involves increase in size and number of cells, while differentiation is about cells becoming specialized to perform particular roles. As this development unfolds, a complex body organization begins to take shape. We see roots that anchor and nourish the plant, leaves for photosynthesis, branches for support and transport, flowers and fruits for reproduction, and seeds for future growth. Eventually, of course, these structures reach the end of their life cycle and die. This entire process highlights how dynamic and intricate plant development is, connecting back to the broader theme of understanding how plants grow, develop, and reproduce successfully. Let's take a closer look at the concept of growth. On this slide, we see that growth is fundamental to our success, and it's not just about increasing numbers or expanding operations. It's about developing sustainably and building resilience over time. When we talk about growth, we're also referring to innovation, improving our processes, and expanding our market reach. It's the driving force behind staying competitive and relevant in our industry. Remember, growth should be strategic, aligned with our overall vision and goals. By doing so, we can ensure that every step we take contributes meaningfully to our long-term success. This slide highlights that growth isn't just a goal. It's a continuous journey that requires focus, adaptability, and a clear understanding of our key drivers. Growth may be defined as an irreversible, permanent increase in size, volume, or mass of a cell, organ, or even an entire organism. It's one of the fundamental characteristics that make living beings unique. When we talk about growth, we're referring to these tangible changes that we can observe, like a seed turning into a plant or a child growing taller. It's important to understand that growth is always accompanied by metabolic processes. These processes include anabolic activities, where building blocks are assembled, and catabolic activities, where larger molecules are broken down. Both of these processes happen at the expense of energy, which fuels the growth. For example, think about the expansion of a leaf or the elongation of a stem. These are simple, everyday illustrations of growth in plants. They highlight how growth involves physical changes in size and form, driven by complex biochemical activities. Overall, growth is fundamental because it reflects the vitality and development of living organisms, connecting directly to their ability to adapt, reproduce, and survive. It's a vital sign of life itself. Let's take a closer look at the main characteristics of growth. First, cellular growth involves not just the increase in cell size, but also processes like cell expansion and differentiation. Cell division is another key aspect, where one cell divides to produce new cells, supporting growth and development. Growth rate, on the other hand, can be defined as the amount of growth that occurs per unit of time. We can even express this mathematically to analyze growth patterns more precisely. Overall, understanding these fundamental characteristics helps us grasp how organisms develop and adapt over time, tying into the broader theme of biological growth and regulation. Now, let's take a closer look at some key concepts related to growth. First, we have the base of natural logarithms, which we denote as e. This number is fundamental in describing exponential growth. Next, t represents time of growth, and r is the growth rate, a measure of how quickly something is growing over that time. We also define W0 as the initial size at the beginning of the period, whether that's weight, height, or some other measurement, and W1 as the final size after growth has occurred. The relationship between these is expressed by the equation W1 equals W0 times e to the RT power. This exponential growth model shows that if you plot the growth parameter against time, you typically get a sigmoid or S-curve. Think about biological systems. Cells, tissues, and organs all tend to grow this way. Initially, growth is slow during the lag phase. Then, it accelerates rapidly during the exponential or log phase. Finally, as resources become limited, growth slows down in the stationary phase. Moving on, let's consider geometric growth. Here, the growth rate r is defined as the elongation per unit time. We track length over time, where L0 is the length at 0 time, and Lt is the length at some later time t. The relationship is linear. Length at time t equals initial length plus r times t. Mathematically, this is expressed as Lt equals L0 plus r times t. An example would be a root elongating at a constant rate. After cell division, only one daughter cell continues to divide, while the others differentiate or mature, which influences the growth pattern. Finally, we have arithmetic growth, which is a different concept altogether. It's characterized by a constant addition over time rather than exponential or geometric patterns. Understanding these different types of growth and their rates helps us describe and predict biological development more accurately, tying into the overall theme of growth dynamics in living systems. Let's take a closer look at differentiation, dedifferentiation, and redifferentiation. These are fundamental processes in cell development and tissue regeneration. Differentiation is when a cell becomes more specialized, developing specific functions and characteristics. It's like a stem cell choosing a particular career path. Dedifferentiation, on the other hand, involves a specialized cell reverting back to a less specialized, more primitive state. This process is crucial during tissue repair and regeneration, as it allows cells to regain the ability to divide and then redifferentiate into the needed cell types. Redifferentiation is simply the process where these dedifferentiated cells once again acquire their specific functions, completing the cycle. Understanding these processes highlights how flexible and dynamic cells can be in response to injury or developmental cues. They're essential in both normal development and healing, and they connect to the overall theme of cellular plasticity and regenerative medicine. Cellular differentiation is a fascinating process where a less specialized cell transforms into a more specialized cell type. Think of it as a cell gaining new skills and functions that are specific to its role in the plant. Cells that come from meristems and cambium are initially quite versatile, but as they differentiate and mature, they become highly specialized to perform specific functions. This process of differentiation is crucial because it allows plants to develop complex structures and carry out diverse functions effectively. So, in essence, differentiation is how cells evolve from being generalist to specialist. ensuring the plant's growth and functionality are precisely tailored to its needs. Cells undergo significant structural changes during differentiation, and these changes happen both in their cell walls and their protoplasm. For example, when forming trachyry elements, cells actually lose their protoplasm, which is a key step in their development into specialized water-conducting cells. Plants, on the other hand, develop very strong and elastic secondary cell walls that are rich in lignocellulosic material. These thickened walls are essential because they allow the plant to carry water over long distances, even under extreme tension. This whole differentiation process is crucial because it enables cells to specialize and perform specific functions, which ultimately supports the plant's overall growth and survival. So, understanding these structural changes helps us see how plants adapt and optimize their water transport system. Let's talk about dedifferentiation, which is a fascinating biological process. Essentially, it's when cells that have become specialized or differentiated lose some of that specialization and revert back to a more primitive, stem-like state. This means they regain the ability to divide again, even if they previously couldn't. This process is particularly important in plants, especially during secondary growth, where it helps in forming new tissues. It's also a key part of wound healing, allowing plants to regenerate damaged parts by reprogramming mature cells. So, dedifferentiation highlights the incredible plasticity of cells, showing us how they can adapt and revert to a more versatile state to support growth and repair. It ties into our overall understanding of cellular flexibility and regeneration across different organisms. A dedifferentiated cell has the remarkable ability to divide and produce new cells. These new cells, however, don't stay in a dividing state forever. After dividing, they lose their capacity to divide again and become part of what we call permanent tissue. This process is known as redifferentiation. It's a crucial concept because it shows how cells can revert to a more specialized state, or alternatively, how they can contribute to the formation of abnormal growths, like tumor cells. For example, in the formation of tumor cells, dedifferentiated cells can proliferate rapidly, but then they often undergo redifferentiation, which can lead to the complex behavior of cancers. So, understanding redifferentiation helps us grasp how cells maintain tissue stability, or, conversely, how they contribute to disease processes. Let's move on to the development phase. This is a crucial part of the process where we see ideas transforming into tangible results. Here, we focus on refining our concepts, ensuring they align with our goals, and making sure they're feasible to implement. Development is about turning potential into reality through careful planning and execution. It's also where we identify challenges early on and adapt our strategies accordingly. Remember, strong development sets the foundation for success in the later stages. Throughout this phase, collaboration and feedback are key, helping us stay focused and on track. Connecting this to the bigger picture, effective development ensures that our vision becomes a practical, impactful solution. Let's take a closer look at the sequence of the developmental process in a plant cell. Development, in this context, refers to all the changes that an organism experiences throughout its life cycle. This spans from the moment a seed germinates, beginning its growth, all the way to senescence, which is essentially the aging process. Understanding development is key because it helps us see how cells grow, differentiate, and contribute to the overall function of the plant. Throughout this presentation, we've been emphasizing how complex and coordinated these processes are. So, when we talk about development, think of it as a series of carefully regulated steps that transform a simple seed into a mature, functioning plant. Let's take a closer look at an interesting phenomenon called heterophyly. This is when a single plant can produce different types of leaves on the same plant. For example, in cotton, coriander, and larkspur, you'll notice that the juvenile plants have leaves that look quite different from those on mature plants. It's like the plant changes its leaf shape as it grows. This ability to produce different leaf forms is part of what we call plasticity. Essentially, plasticity is the plant's capacity to adapt and modify its structures in response to environmental conditions and different stages of its life cycle. It's a remarkable feature that helps plants optimize their growth depending on their surroundings. Now, when we talk about plasticity and heterophily together, we're highlighting how flexible plants can be. They can follow different developmental pathways, producing a variety of structures to better suit their needs. This adaptability is a key part of how plants survive and thrive in diverse environments. So, in the context of our overall theme, understanding plasticity and heterophily helps us appreciate the dynamic ways plants respond to their environment and developmental cues, which is fundamental to plant biology and breeding strategies. Now, let's move on to plant growth regulators. These are substances that influence the growth and development of plants in various ways. They are crucial tools in agriculture and horticulture because they help manage plant behavior, improve yields, and even control plant shape. Think of them as the plant's own chemical managers, directing growth processes to meet specific needs. They include hormones like auxins, gibberellins, cytokinins, abscisic acid, and ethylene, each playing distinct roles. For example, auxins promote root development and influence fruit growth, while gibberellins are known for stimulating stem elongation. Cytokinins are involved in cell division and shoot formation, and ethylene is key for fruit ripening and response to stress. Understanding how these regulators work helps us better manipulate plant growth for optimal results. This ties into our overall theme of how controlling plant processes can enhance productivity and plant health. Plant growth regulators, also known as phytohormones, are chemical substances that play a crucial role in influencing how plant cells, tissues, and organs grow and differentiate. Think of them as messengers that facilitate communication within the plant, ensuring that various parts develop in harmony. These regulators work together to coordinate the growth and development processes across different cells, maintaining the plant's overall health and functionality. They come in various chemical forms. For example, some are indole compounds like indole 3-acetic acid, or IAA, which is a type of auxin. Others are adenine derivatives, such as kinetin, which promote cell division. There are also derivatives of carotenoids like abscisic acid or ABA that help plants respond to stress. Additionally, some are terpenes, such as gibberellic acid, GA3, which stimulate elongation and growth. And finally, we have gaseous regulators like ethylene, with the formula C2H4, which influence processes like fruit ripening and senescence. Understanding these diverse plant growth regulators helps us appreciate how plants carefully regulate their development, ensuring they grow properly and adapt to their environment. This interconnected system of chemical signals is fundamental to the overall growth and productivity of plants, tying directly into the broader theme of plant development and regulation. Now, let's take a closer look at some of the synthetic auxins. We have 24D, which stands for 2,4-dichlorophenoxyacetic acid. This compound is widely used in weed control due to its strong herbicidal properties. Then there's NAA, or naphthalene acetic acid, which is commonly used to promote root formation in cuttings. IBA, or indole butyric acid, is another important synthetic auxin often applied to stimulate rooting as well. And finally, IAA, or indole acetic acid, is actually the natural auxin found in plants, but it's also used in synthetic form for various growth regulation purposes. Auxins as a group were actually discovered by Charles Darwin and his son Francis, who observed their effects on plant growth. Later, FW went isolated auxins from the tips of coleoptiles. Those are the protective sheaths covering young grass shoots, specifically from oat seedlings. He developed a bioassay called the Avena curvature test, which allowed scientists to measure auxin activity precisely. When we talk about plant growth regulators, or PGRs, auxins are a key category. They play vital roles in cell elongation, root initiation, and overall plant development. Understanding these synthetic auxins helps us harness their power for agricultural and horticultural purposes, making them a crucial part of our plant growth toolkit. Now, let's take a closer look at some of the key physiological effects of auxins. These plant hormones play a crucial role in controlling xylem differentiation, which is essential for the plant's water transport system, and they also help regulate cell division, supporting overall growth. One interesting application is their ability to induce parthenocarpy, meaning fruit development without fertilization, such as in tomatoes, which can be very beneficial for certain agricultural practices. Auxins also promote flowering, as seen in crops like pineapples, encouraging earlier or more abundant flowering. Additionally, they are used as herbicides, which might sound surprising, but certain synthetic auxins can selectively target weeds. They also promote abscission, meaning they help the plant shed older, mature leaves and fruits when it's time to do so, aiding in plant health and harvest management. Another important role is in initiating rooting in stem cuttings, making propagation easier for growers. Auxins can also prevent premature fruit and leaf drop during early stages, helping plants retain their valuable parts longer. They even promote apical dominance, which is the growth of the main shoot over side branches, shaping the plant's overall form. All these effects highlight how versatile auxins are in plant physiology and agriculture. They're part of a broader classification of plant growth regulators, or PGRs, which include other hormones with different functions. Understanding these roles is key to effectively managing plant growth and productivity in various agricultural and horticultural contexts. So, a significant discovery was made by E. Kurosawa, a Japanese scientist, who identified gibberellins in a fungal pathogen called Gibberella fujikuri. What's interesting here is that these gibberellins weren't just found in the fungus itself, but they were actually isolated from infected rice seedlings. This was done by treating the seedlings with sterile filtrates of the fungus, which means the fungus secreted these growth-promoting substances into the environment, influencing the plant. This discovery is crucial because it links fungal pathogens directly to the production of plant growth regulators, or PGRs, specifically gibberellins. It helps us understand how certain pathogens can manipulate plant growth, which ties into our overall discussion on the classification and role of PGRs in plant development. Today, I'll be discussing some of the key physiological effects of gibberellins and how they influence plant growth and development. First, gibberellins can induce parthenocarpy, which is the development of seedless fruits. This is particularly important in commercial fruit production because seedless varieties are often preferred by consumers. Next, they play a role in vernalization, which is the process of exposing plants to a period of cold to trigger flowering. This is crucial for certain crops that require winter chilling to produce flowers successfully. Gibberellins also promote bolting in rosette plants like cabbage and henbane. Bolting is the rapid elongation of the flowering stem, which signals the plant to transition from vegetative growth to flowering. Furthermore, they are involved in seed germination by breaking seed dormancy, allowing the seed to sprout and grow into a new plant. They can also stimulate the formation of dwarf shoots, which are shorter, more manageable plant forms. In addition, gibberellins are used to promote flowering in long-day plants, which need longer daylight hours to flower. They can also overcome the natural dormancy of buds, tubers, and seeds, helping plants to grow and reproduce more reliably. Another important effect is the elongation of intact stems, which can improve plant height and overall vigor. All these effects highlight the diverse and vital roles that gibberellins play in plant physiology, especially in agricultural practices, to enhance productivity and crop quality. Finally, this slide touches on the classification of plant growth regulators, or PGRs, of which gibberellins are a key example. Understanding these classifications helps us better utilize them for specific growth responses. Skoog and Miller were pioneering scientists who made a significant discovery in the world of plant growth regulators. They identified a substance called cytokinins, which are crucial for cell division and growth in plants. To study this further, they managed to crystallize the active substance responsible for promoting cytokinesis, which is the process of cell division. They named this substance kinetin, and interestingly, they isolated it from natural sources like corn kernels and coconut milk. This discovery was a breakthrough because it helped us understand how plant growth can be regulated and manipulated. When we talk about cytokinins, we're referring to a specific class of plant growth regulators that promote cell division and differentiation. It's important to see how this fits into the larger picture of plant development and growth control. Understanding cytokinins helps us appreciate the complex hormonal balance that governs plant life, and it underscores the significance of plant growth regulators as a whole in agriculture and horticulture. Now, let's take a closer look at the physiological effects of cytokinins. One of their key roles is to increase cell expansion, particularly in dicot cotyledons and in leaves. This means they promote growth by helping cells grow larger, which is essential during early development stages. Cytokinins also delay the senescence of leaves and other organs, effectively extending the lifespan of plant tissues and maintaining their vitality for longer periods. Additionally, they promote chloroplast development and chlorophyll synthesis, which are crucial for photosynthesis. This not only enhances the plant's ability to produce energy, but also improves overall plant health. Cytokinins also encourage lateral bud formation, leading to more branches and a bushier plant, which can improve flowering and fruiting. Furthermore, they induce cell division, a fundamental process that supports growth and tissue formation. Altogether, these effects highlight how cytokinins are vital regulators of plant growth and development, working in concert with other hormones to optimize plant vitality and productivity. Cousins confirmed that ripened oranges release a volatile substance, and interestingly, this substance speeds up the ripening process of stored unripened bananas. What they found was that this volatile compound is actually ethylene. Ethylene is a plant hormone that plays a crucial role in regulating various growth and developmental processes, especially fruit ripening. When we talk about plant growth regulators, or PGRs, ethylene is classified as one of the key natural regulators because of its significant influence on ripening. Recognizing ethylene's role helps us understand how plants naturally coordinate their growth and how we can potentially manipulate these processes for better crop management. This ties back to our overall theme of understanding how plant hormones and regulators work together to control plant development and improve agricultural practices. Today, I want to talk about the various physiological effects of ethylene on plants. Ethylene is a crucial plant hormone that influences many aspects of plant growth and development. For example, it initiates flowering in pineapples, which is essential for synchronizing fruit set and ensuring a uniform harvest. It also plays a role in breaking seed and bud dormancy, helping plants to start their growth cycle when conditions are right. Ethylene promotes root growth and the formation of root hairs, which are vital for water and nutrient absorption. Additionally, it induces fruit ripening, a process that is carefully coordinated to improve fruit quality and harvest timing. Interestingly, ethylene is involved in sprouting of potato tubers, enabling them to grow and develop into new plants. However, ethylene also has inhibitory effects, such as inhibiting the elongation of stems and roots, which can help plants allocate resources more effectively during certain stages. It also initiates germination in peanut seeds, kickstarting the growth process. Lastly, ethylene promotes senescence and abscission, which means it helps in the aging process of leaves and the shedding of old or damaged plant parts. Overall, ethylene is a versatile hormone that manages key transitions in a plant's life cycle, aligning growth, development, and even aging with environmental cues and internal needs. In 1963, Frederick T. Adicott and his colleagues made a significant discovery by identifying abscisic acid. This was a breakthrough in understanding plant hormones. ABA, as it's commonly called, was actually isolated from various substances that promote abscission, or leaf and fruit drop, in cotton plants. This finding helped us understand how plants regulate their responses to environmental stresses. In fact, ABA is often referred to as the stress hormone because it plays a crucial role in helping plants cope with drought and other stresses by signaling them to conserve water and close their stomata. So, recognizing ABA's role not only deepens our understanding of plant growth regulation, but also highlights its importance in stress responses within the broader context of plant hormones and growth regulators. Abscisic acid plays a really crucial role in various aspects of plant development. It's especially important when it comes to seed development, maturation, and dormancy. Essentially, it helps ensure that seeds develop properly and stay dormant until conditions are just right for germination. One of the key functions of abscisic acid is to stimulate the closure of stomata. This is vital because it helps plants conserve water during stress conditions like drought. By closing the stomata, the plant reduces water loss and can survive tough environmental conditions. In addition, abscisic acid inhibits seed germination. It acts as a signal to keep seeds in a dormant state until the environment is favorable for growth. This is a critical survival mechanism, ensuring that seeds don't sprout when conditions are harsh or unpredictable. It also induces seed and bud dormancy, which helps protect the plant during unfavorable periods, like winter or drought. This way, the plant can pause growth and resume when conditions improve. Lastly, abscisic acid acts as a general plant growth inhibitor. It's involved in balancing growth and stress responses, making sure the plant conserves resources and stays resilient during challenging times. So, overall, abscisic acid is a key hormonal player that helps regulate development, stress responses, and survival, tying into the bigger picture of how plants adapt and thrive in their environments. Now, let's delve into photoperiodism, which is a fascinating aspect of how plants respond to the length of day and night. Photoperiodism influences critical processes like flowering and flowering time, allowing plants to adapt to their environment effectively. Essentially, plants have evolved to detect changes in day length, which helps them know when to initiate flowering, ensuring they do so under optimal conditions. This response to photoperiod is mediated by specific photoreceptors that detect light cues, and these signals are integrated to trigger physiological changes. The key concept here is that different plants are classified based on their photoperiodic response. Some are long-day plants, others are short-day plants, and some are day-neutral. Understanding photoperiodism is vital because it connects to the overall theme of how plants adapt and optimize their growth and reproduction in relation to environmental cues. It highlights the incredible ways plants interpret subtle changes in their surroundings to ensure survival and reproductive success. Photoperiodism is essentially how plants respond to the relative lengths of light and dark periods they experience. It's fascinating because some plants need a certain amount of light exposure to trigger flowering, so their development depends on these environmental cues. But it's not just about how much light they get. The duration of the dark period is equally important. In fact, the length of darkness can determine whether a plant flowers or not. This concept helps us understand how plants adapt to their environments and synchronize their growth cycles with seasonal changes. It's a perfect example of how environmental factors influence plant behavior, fitting into the larger picture of plant physiology and adaptation we've been exploring. Now, let's take a closer look at some interesting examples of plant responses to light and temperature. For instance, plants like cucumber, rose, and tomato are what we call day-neutral plants. This means that their flowering isn't really influenced by how long the day is or the amount of light they receive. They essentially flower regardless of the light duration, which makes them quite adaptable. Moving on, we have plants such as rice, jawar, and cotton. These are examples of what we term plants requiring a specific exposure to light, but only for a period less than a certain critical duration to induce flowering. Their flowering depends on reaching that optimal light exposure, making them sensitive to photoperiod conditions. Another fascinating concept is vernalization. This is a process where plants need to be exposed to low temperatures to induce flowering. Vernalization actually helps prevent plants from flowering too early late in the growing season, giving them enough time to mature properly. For example, biennial plants like sugar beet, cabbage, and carrot are monocarpic, meaning they flower once in their lifetime and then die, often requiring vernalization to trigger flowering at the right time. Then we have short-day plants, which require a certain minimum length of darkness to flower. They usually flower when nights are longer than a critical period. Conversely, Long day plants need exposure to light for a duration longer than a critical period to induce flowering. Examples of these include peas, barley, and wheat. Understanding these different plant types, day-neutral, short-day, and long-day plants, helps us grasp how plants adapt their flowering based on environmental cues. It's all about how they perceive and respond to light and temperature, ensuring their reproductive success. This knowledge is essential for crop management and optimizing flowering times in agriculture.